Check, check. 1, 2, 1, 2. Check, check. 1, 2, 1, 2. Intro, check, check.
Testing, 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 testing.
Unmuted.
just about 12 now. So welcome to Applied AI Lunch Talk. Welcome here to Skylab and also to the people who are joining us online. I know we have a crew sitting at the Pioneer Center for AI enjoying some pizza as well. So I'm super excited to be able to host this event where we've chosen to put the spotlight on biotech and the advances that AI can facilitate in that realm. With me today, I have two speakers. They all have their origin at DTU, and they are biotech founders. So the first one I'm going to welcome on stage is Christopher Workman from Tailwind Biotech, and the second speaker is going to be Bogomil Simon from Faro Bio. So I'm really happy that they're able to join. I know it's been something a lot of my colleagues have also been looking forward to here at DTU. So I think without further ado, I'm just going to hand the stage and the clicker to you. And please take it away. Give him a big hand.
Yes, first, thank you to the organizers for inviting me and inviting me again after I was not able to make it the first time. But thank you all for coming. So I'll just very briefly give you a short CV on me. So I'm originally a molecular biologist, educated at University of Colorado in Boulder, and I studied computer science after that because I got very interested in the Human Genome Project and all the things that I could see were needed to bring value to these genome projects. So I got very interested in bioinformatics, came to DTU, did a PhD in bioinformatics, and then did a postdoc in San Diego, came back to DTU to start a research group. So I have about 17 years of research as a faculty at DTU, and I know some of you from that time as well. So that's a brief background on me. And lately, I've been focused on my own startup that I founded, and some of the technology, of course, came out of the research that my group was doing at DTU. So the talk is in roughly two parts. So first, I'm going to motivate the subject to some best I can, and it's going to have a biotech feel, and it's going to talk a little bit about some things you may or may not be aware of in how we create solutions, technical solutions for challenges that we have in society, and some things that may have come to your awareness through popular culture, and as you'll see. So the background here is some of the technologies we are seeing today come from these drivers of the technologies. So sequencing DNA has been becoming more and more affordable, as you know, and you can see this graph goes back to 2002, so it's over more than 20 years of technological progress, and we can see the costs have dropped off more than exponentially, in particular here in the early 2000s, mid-2000s. And now, of course, we can sequence a human genome for much less than $1,000, and that's actually was seen as a very difficult task not so many decades ago. But alongside that, alongside the sort of ubiquitous and relatively cheap DNA sequencing that we can also do DNA synthesis. So these are tracking maybe not as fast, of course, as sequencing, but the ability to create de novo DNA sequences. In fact, there are companies out there that are synthesizing whole genomes for microorganisms. There have been projects to re-synthesize the genome of yeast as well, so actually even larger genomes. But nowadays, we can synthesize DNA nearly as effectively as we can sequence it. You can see the costs here are still higher, though. It costs more in materials to do the synthesis. So we can read and write DNA, and we would argue as biotechnologists that our ability to read and encode information in DNA is in fact developing faster than our advances in hardware and integrated circuits. So that is something that I think is still going on. This has been going on for a few years now, and how that can find its way into solutions in everyday life. We're starting to see more and more of that, and I know there are some companies that are offering data, long-term data storage as DNA. That is actually now becoming more viable. That in fact, DNA is a very excellent high-density information storage medium. Where have we come from? So basic DNA technologies and genetic engineering, and now synthetic biology, came from, and I show you these pictures. This is where we came from as biotechnologists, this cut, copy, paste, where we take pieces cut them out from natural sources, and we reorganize them, and we make new things, new technologies based on this cut and paste of DNA. So that has gotten us far. We have developed lots of drugs. We've solved, in large part, many hard problems with regard to getting cells to make things for us to treat us, and such as diabetes and insulin that we all know is really important for the Danish biotech economy. But today we think about this more as synthetic biology, and with synthetic biology we add on this extra technology of design, that we can design new things that we've never seen in biology before. And of course many of you are possibly aware that many companies out there are designing primarily protein sequences, new proteins that fold in ways that we've never actually seen in nature. And I'll talk briefly more about that. But the synthetic biology field from which, and this is still coming from primarily a cut, copy, paste approach. We've been able to engineer full pathways into organisms. So here we're making a chemotherapeutic, a very complicated molecule called Taxol, or paclitaxel. Some of you may have heard of this. It's used for treatment of breast and ovarian cancer, and something that I also worked on. My first jobs was to isolate Taxol, and it's in very, very low concentrations in Taxus brevifolia. It's a yew tree, Pacific yew tree. And so now it can be made either semi-synthetically from engineered organisms. Another one is the vinblastine story. This has some connection to DTU. It's from a number of years ago, where they've engineered microorganisms to produce a drug, vinblastine, which, as you can see, is a very complicated molecule. So this is something biology is very good at doing, and we are now able to, in some extent, say we want to make this molecule. What are all the steps we need to engineer into a microorganism to make that molecule? From a few years back as well, this got some attention, and you may have seen this, basically brewing things like THC. Of course, that is technically possible. Does it replace the naturally occurring ways of getting THC? No, because plants tend to be already very good at doing this naturally, but nonetheless, this raised a few alarms saying, okay, now people are going to grow this at home. This maybe was around the time lots of states were legalizing use of marijuana anyway, so we don't hear too much about it today. Another wave that we've seen is that we can engineer organisms to make certain proteins that we can add to food. So this is leg hemoglobin. So it's a plant hemoglobin. It's not actually an animal hemoglobin, but it's a plant hemoglobin that we've been able to add to essentially veggie burgers to make them more meat-like. And sure, many of you have experienced that. As well as things like replacing, instead of sourcing silk from silk moths, we can actually engineer microorganisms to produce those proteins in ways that we can simulate that textile. So this is now stepping into what we would call biomaterials, bio-based materials, which is also an area that's growing. And of course, it should be, in principle, far more sustainable than farming these animals. This is more speculative, but there is a company that is trying to revive through de-extinction, so to revive the woolly mammoth. They have some arguments for why this could be important for, say, Siberia and maintaining ecosystems. And that technically, if we can synthesize a yeast genome, it shouldn't be too many years before we are able to partially synthesize higher eukaryotes such as mammals, but I would still say we're still fairly far off. We're in the press very recently. This was from a few days ago, where they were showing they could make a woolly mouse. So this is, there's some debate as to whether this is relevant for turning an elephant into a woolly mammoth, but nonetheless, they could show that they have some way of controlling the phenotypes of fur in mammals. So the woolly mouse has been given to us. I will switch a little bit here towards, this is more of a general slide showing that how we do this in practice in not the mouse, but anyway, in when we make proteins in a lab, we typically do this in large tanks. It's a process very much like brewing beer. We do a fermentation in a large tank, and there are many steps upstream, so this is called the upstream, and then also many steps downstream of the fermentation. So this is where sort of the products are made, but then of course with regard to drugs, there are many steps downstream that are important, as well as many steps upstream. So the company, our startup, which I'll tell you about, we come in very far upstream, where we apply some AI, and I'll tell you more about that in a few slides, where we design the DNA that goes into the cells, we engineer into the cells, which hopefully you can see we are able to do, and then hopefully we can address many different types of bioproduction problems with what we're doing with our AI. You're probably more familiar with how AI is applied to the concept of protein design. So this was out not so long ago. This large language model applied to go from sequence to structure and function with regard to proteins. And I think one of the things that I will sort of focus in on here is the scale of these models, as you may or may not be aware of, has on the order of 100 billion parameters, and it's trained on about 3 billion proteins. So you may, from a classical AI perspective, we are basically memorizing, to some extent, all the protein structure data that is out there, and then we're able to use that, even though we're very likely overfitting quite a bit, we're still able to use that to present these models with sort of partial prompts of whether it's the sequence structure or function pieces missing, and then it can, of course, give you the most likely prediction as to what amino acids belong in the rest of the sequence. So there's a whole set of companies who are trying to do just this, to engineer novel proteins for all kinds of different applications. This is fairly new, but I won't talk about large language models. We don't use large language models. Large language models, of course, require very large corpuses of data, but Tailwind, we say we are using AI to build intelligent DNA sequences. And first, maybe we'll have to sort of see where does this technology fit in. When we have a genome, DNA sequence, we need to make some things from that DNA, and there are a number of steps that are needed to get from the DNA to the protein, and it's not meant to be a lesson in molecular biology, but this is just to highlight where we are. We're in this step, the transcription step, so when we go from DNA to the RNA, which is this intermediate messenger that then gets translated into protein. But there, of course, are steps of regulation all along the way, but we are mostly concerned with engineering the DNA that gives us the best transcriptional output. And that means we are focusing on promoter sequences, which I'll tell you about in a moment. So the problem here in biomanufacturing-based companies is that engineering their strains for a new bioprocess takes a lot of time. Maybe they don't have the methods to select the best sequences. There's a lot of trial and error that's going on. That's common practice. And there are many suboptimal bioproduction processes that take many years to debottleneck or to optimize. So, like, for example, the initial groups that had yeast making cannabinoids, the yields are so low. It's usually when you first show that you can get an organism to make something, it's not commercially viable. So there's a lot of technology that needs to be brought to bear to bring these bioprocesses to commercially viable state. And that's where we hope we can do that, help companies get faster to a commercially viable bioprocess. So part of how we do this is just an overview of our method. Is we have a machine learning approach that takes as input genetic data, and this primarily comes from large publicly available genomes. So there are lots of genomes out there that we can use. We are, as a business-to-business company, we talk to the companies and say, what are your challenges? What are your production hosts? So they need to tell us a little bit about what they're doing and what kinds of organisms they use. And then we can go and find publicly available data on related species and extract the DNA. And sometimes we have to annotate these genomes. Many of them are not annotated. So that's part of our process: annotation, collation of data, and then we can build many models for the functional DNA sequences we want to design. So, I mean, that's the way genomes function. There are many sort of cassettes. So each gene has a whole host of functional DNA sequences around them, including or within the coding sequence itself, that make a gene a gene. So we are able to build for the few thousands or 10,000s of genes in an organism, build models for each gene. And at the same time, we're able to identify also through bioinformatics, which of these sequences might be most suitable for the process that a customer is trying to develop. So we have prior gene ranking or prioritization approach at the same time. So then we present candidates to companies, and here's just some of the data we have from our initial studies. So in the black bars, we're seeing protein production as measured by fluorescent proteins, driven by the expression of our design promoter in dark and relative to a native promoter in the cyan. So we were able to generate designs that outperform the natural sequences, and these are known to be strong. It's a fairly commonly used, very strong promoter, strong constitutive promoter, it said, so it should be on all the time, producing a lot of transcripts, a lot of gene expression, a lot of protein. So we're measuring protein and comparing to the natural promoters that generate that protein. We've also moved into, this is Comictylophafi, it's a yeast, and we can see what we have achieved anywhere from 50 to 400-fold, 400% increases over other strong promoters. And this is a very strong promoter in this organism, and we could improve that about 40% or so. So we have shown that our technology works in a number of organisms, including Saccharomyces cerevisiae, which is the standard budding yeast. I don't show you that data here, but these organisms are filamentous fungi that are important to novonesis, and a lot of the enzyme producers use these organisms for production. Our team, we're a small team. We have our CEO, is Mirko, and he's comes with a business background from CBS at KU, and Lorenzo also has a background from Novo and Stanford, and he's helping make usable code out of my academic code, which is very, very good, and doing a very professional job in making us scalable. And we have Professor Henrik Simonsen. He was also at DTU, and he's our plant synthetic biologist, and he's helping us with business development. And of course, I'd just like to thank DTU for helping us get off the ground as a startup, and as well Innovation Fonden and the Danish Agency for Higher Education and Science for support. So I'm happy to take questions if you have any. Thanks.
Thank you for a wonderful talk. Let's see who have questions. Wow, we have an excellent audience today. Yes.
Thank you so much. I'm a little bit curious about some of the. You said that many of the products that are produced through fermentation are not readily usable in the first couple of trials. And I would like to hear more about which issues you're actually facing and how you're going to solve those. Yeah.
Yeah, so many times people have an interesting problem. They find some technical solution for that problem, but it's not commercially viable for the reason that the yields are too low or their costs relative to what they can sell is too high. So that usually happens when you are engineering a bioprocess. So the first number of iterations, it won't be viable for reasons that you're trying to make the cells do something they're not naturally interested in doing. It's not a benefit to them to make what you want them to make. So it's about engineering clever things like what Enduro's doing, circuits that addict the organism to the products it's making. You trick them into producing more of what you want them to produce. Also, there are lots of competing pathways that might be taking. So if you have 6 steps in a pathway and some intermediate concentrations you want to be rather high are getting siphoned off into other competing pathways. So you would like to downregulate those pathways. So it's like, think about plumbing and valves, and you have flows through these networks, and you'd like to optimize the flow to your product. So there's some aspect of engineering, genetic engineering the organism to maximize the flux of flows into the products you want. Sometimes the products make the cells sick that you're getting them to make. That often happens, so you can use adaptive evolution to maybe also allow the organism to learn how to produce high concentrations of a product that would otherwise inhibit its growth. Also getting the product out of the cells, so the export of the product, export of the protein can also be a bottleneck, a challenge. Does that partially?
Yeah. The last one covered functionality a little bit. Are you covering functionality in any other aspects? The functionality of the product?
We are not. So we are focused on and maybe I haven't made that entirely clear enough in my talk, but we're functioning on the regulatory sequences that control the output of the genes that's like the first step in getting to the protein. Can be a rate limiting step that if you don't get the mRNA made, you don't get the protein. So we're focused on pushing up the levels of mRNA higher, and then what you saw from the bar charts was that we actually could do that and get the protein levels higher as well. So, yeah.
Thank you.
Show of hands, who else have a question? Yeah, coming behind you.
Thank you. I think as a follow-up question to that one, why are you only interested in the promoter sequence and the regulatory sequences than the gene or enzyme sequence itself? Because I think that's where it's like more, more popular, let's say.
It's certainly more popular to focus, and when we are going to have DNA synthesized, the assumption is you will be synthesizing a coding sequence. So we're not doing that. Part of it is we're trying to meet a need that isn't well met. So there are not many companies out there doing exactly what we do. We had the technology to meet this need that we developed at DTU. So that's part of the reason we're trying to focus. When we started on this journey, I was thinking early on, I want to do this, all these things. And immediately someone said, nope, just focus on this. And as an academic, that's a little bit hard to do, I have to say. I would have loved to. And we do offer codon optimization. We can do that. Yes, we can do codon optimization for sure. Designing new protein sequences, that's not something we have focused on. But there are many companies that do that. So I think it's a highly competitive field right now, I would say.
Thank you.
Do we have more questions? Any online, perhaps?
Thank you, and that's a question from Ruth Castro Silva. And she's asking, she's saying that there's been some concern around DNA data sourcing. So with the AI added to this equation, do you have any concern about the future of DNA AI and how it will power research?
So if I understand the question about, there are, the synthesis companies also screen to make sure you're not sending in some pathogenic sequence, that you're not going to try to make, reconstruct the poliovirus or things like this. So if that's the nature of the question, we are not using any coding sequences in what we build our models on. So by definition, the sequences we use and the sequences we generate can't be used they can be used for lots of things, but they themselves are not, could not be seen as a biohazard. If that makes sense. So they should be within the safe practice guidelines. I hope that answers their question.
I hope so too.
But the companies are, of course, very interested in making sure they screen and not accept in their synthesis pipelines anything that can be used for dual use. Yeah.
And if it doesn't answer the question, Ruth, you're welcome to write something again and clarify your question. Any other questions in the room? Yeah.
I have one question myself, because now we are at DTU Skylab, where we are looking very much into how we can foster innovation and make more startup researchers becoming startups spinning out of the university. And you have been on this journey yourself, so I'm a little bit curious to hear about how that has been to go from research and into working in a company. And if you ever dream of going back.
Actually it was fairly easy. After 17 years, I think having a change in your career focus is not a bad idea. So from that perspective, it gave me something fresh and new to a way of thinking about problems I was approaching. The process at DTU they've been very supportive. So the mentor program we were in, and we were also here as a startup at Skylab. So that was it's all been made it easy for us. But it's hard to run a research group and build a startup company unless you really have someone take full charge of the project outside of DTU. That I think it would be hard. Not that I've tried to do both. Well, maybe we did. I tried to do both for a while, and we didn't really launch the company. Is basically what happened. Yeah, too busy.
Do we have another question in the room? Otherwise, I have one. I'm curious about your next steps in Tailwind Biotech. What do you need next? Do you need customers? What kind of profiles should they have? Do you need investors? What's your next big challenge?
Yeah, we've been speaking to some companies of very different sizes, some very big companies as well. We have some customers have been small companies so far, and companies that are developing a new process, and ideally in yeast or filamentous fungi, because that's where we've really proven our technology. We do have a lot of companies approaching us about engineering bacteria, bacillus or E. coli. I'm sure we could do something, but the space is fairly saturated for DNA sequences and what they can use. But nonetheless, we can do that. Some big companies like to do everything in-house, so they don't believe they need us, which is a bit of a shame. But anyway, we're looking for companies that are in the medium size who are developing new technologies and are willing to test out, try new cutting edge technologies. So
cool. Well, thank you so much. I think this was a wonderful. We have one more. We'll squeeze it in.
I just thought of this. I was wondering, your product line. So, are you developing promoters that are organism specific, meaning that you only are limited by that organism, or is it depending on the sequence? Like, is it like specific to the DNA sequence to optimize for that production? And I ask this because are you able to reuse the products that you're making, or is it specific to the application every single time?
You should be able to reuse them. I mean, our business model has changed a little bit where we're now focusing more on selling our designs, but initially we would like to license out to many customers the same designs, the same promoter sequences. They tend to be, I wouldn't say, they tend to be genus specific. So it should work within the genus. Usually a genus is, there are not that many production organisms from, Aspergillus might be one exception. There are 200 genomes for Aspergillus that we know. But there's a small handful of Aspergillus organisms that are used. And so our design should work in Aspergillus in general. So somewhat specific, but not only one organism. That's a good question. That's a very good question. Yeah.
So I was thinking, I'm coming from bioinformatics, and so how do you approach, like, do you patent your technology, or do you keep it a secret, or like, because software can be difficult, and I guess you also published some of your academic work. Okay. So how do you go from that into a business?
So the method is what's the subject of the patent, and it should be granted here fairly in the next few months in the US and in Europe. So right now it's the method that's being protected. We initially wanted to patent we can patent the sequences. Part of the challenge is we would like to patent a library of sequences, and patents don't usually work that way. They want you to focus on this idea of unity. So a collection is not something that's easy to show as a unity of invention. So they want you to patent each sequence. So the cost there could get high very, very fast. But we did, right now we're trying to keep the design secret. The customer can choose to patent if they want, if we sell them the design. So it's a little bit like selling the IP, to be honest. So there should be higher prices for exclusive sales of designs relative to licensing to multiple customers. So that's how we're doing it now.
Thank you.
Thank you so much, and for paddling a little bit for us here. I'd love to say thank you to you, and welcome to you.
You need this. Yeah, exactly. Thanks. All right. Hello, everyone. My name is Bogumił, and I'm leading Ferros Bio. And I have a couple of videos in there, so if something doesn't work, don't kill me. That's an experiment of making it a little bit more inspiring and engaging. All right. So a light bulb is a symbol of human ingenuity. And we invented it 100 years ago, and we didn't stop innovating. So we've built cars, we've built planes, and we also conquered the moon. And this constant urge to exchange ideas, innovate, made us to build a computer and the internet. And in biotech, we also were not left behind. 50 years since the discovery of double helix, we sequenced the complete human genome and also learned how to modify it using CRISPR-Cas proteins. However, one challenge still remains, where we are really slow and inefficient. Drug discovery cycles take more than 10 years, and 90% of drugs never reach the market. It's a big issue, and it highlights a couple of problems, mainly slow R&D cycles. And partially they can be attributed to long wet lab validation, but the larger issue at hand is how we come up with ideas, experiment, and strategies. These days, we face a new tech revolution. AI went from beating Garry Kasparov in chess to solving protein folding challenge using AlphaFold. And this can partially be explained through increase in computational power, but also increase in dataset sizes. Biological data is vast, and scientists these days analyze it using manual tools. We surveyed dozens of researchers, and they mostly reported Excel, Prism, Benchling, Python, or R. Unfortunately, there's just too much information out there to analyze it manually. One example of this is when you look at the number of publications released every year, there's over 3 million. Let's assume 1 million is relevant for a given researcher. If a researcher can read 3 papers per day, he will access 0.1% of the available knowledge. So what it means is that 99% of ideas that the researcher could use will never be reached. And this is only one of the examples of how vast biological information space is. So how do we tackle this problem? Well, we can combine existing knowledge bases that we generated over decades of research with autonomous AI agents that can use biology-specific tools. But so, what are AI agents? How many of you heard this term and feel like you understand what it is? Show of hands. 1, 2, 3, 4. Okay, cool. That's great that I made 3 slides for it. So let's start with a general large language model. You can pose it a question, and if the answer is in the training dataset, then it will provide you an answer. But if it's not there, then you will get some weird hallucination. So we can solve it using compound systems. One example of this is a scientist could ask which mutations of protein X improve binding to the protein Y. And if you ask this question to a general LLM, it will most likely fail. But instead, we can take this query and convert it into a search query against ScamPy database. And if both proteins are there, most likely you will get a correct answer. So we can see this shift from model-centric approach to compound system-centric approach. And this is because these days, the large language models, they have billions of parameters. And even if you retrain them every month, in this month, you will have a multitude of information that you will never have in the model. While compound systems, they are really modular and easier to adapt. But then you might ask, okay, so where is the agent in it? The agent comes with control logic. So normally, we control the flow of the workflow using programmatic approach. So we can make a stringent workflow with if-else statements, and it will decide how the software will analyze the data. And this is very stringent, and it will work if the workflow has to be really robust. But these days, you can put an LLM in the steering of how the workflow will work. So the LLM can decide where to take the flow of the program. And we can consider this as a slow thinking system, because maybe the agent will iterate many times before it reaches the answer, but it's also very flexible. So what properties does the agent has to have? One of them is to reason. What does it mean? It has to be able to plan its course of actions. Another one is to act. So the agent has to be able to use tools. A tool in the industry term would be search for a database, or running a Python script or R script, or using a tool like AlphaFold or Autodoc. And also an agent has to have access to memory. And why is it important? Well, if in the first run an agent writes a script that throws an error, then in the second loop it can correct this error. Okay. So how does it work within the context of biology? I will show you one example that we tried. Imagine you run a large hit screening. You found the right hit, but you don't know yet how it works. So you design CRISPR knockdown experiment, and you have a dataset for it. You can upload this dataset and pose the question, find potential drug targets. An agent will prepare a plan for it, write a Python script that will probably look through your list of hits and fault changes, that will sort them and execute the script and come up with top genes, the genes with the highest fault changes, for example. And this is just first iteration of an agent. What would happen next is probably taking this gene, searching for a database of publications, looking up how this gene could be involved in the disease mechanism and so on. And then the agent can report it. So I will show you how we did it within the platform. The way it works is that scientists can connect all of their databases or upload the spreadsheets directly. In this case, we upload this CRISPR gene knockdown dataset. This is saved, and we can start a new conversation. That's how the dataset looks like. And the scientists can pose a very general question, like find potential drug targets, and set it to auto mode. And in auto mode, the agent will choose the right questions to ask, write a code to analyze the data, summarize it with summary statistic plots, and interpretations. And the interpretation is based both on the table and the vision model that looks at the plot. And what is nice is that the agent also searches through the papers. So when it interprets the data, it uses it, it does it using the literature. And at the end, when a scientist is satisfied, he can take all of those results and summarize them in a report. And this is a fully AI-generated report. And in this report, we can see all of the results, so plots, summary statistics, tables, all of the conclusions, and also references to the literature. And the fun part about this report is that we actually found the same gene as the researchers that provided us with this data set. So we basically recreated part of their research paper in the platform within a couple of minutes. And let me tell you a bit about other features that we're working on. One of them is deeper research. And the whole idea here is that maybe when you use a vector database, you will retrieve first 5 publications that are not so relevant. But based on those publications, normally scientists would go back, learn a bit more about the topic, and continue digging until he has the answers. And that's basically how the deep research works. So we pose an initial question, we search through the database of publications that we vectorized, and then we basically stimulate the agent to decide, do we have enough information? Should we continue searching? And once this is complete, we can either have it as a deep report, or we can do something else with it. We can, for example, use it to extract numerical variables from the publications. So we did an experiment last week with this, where we took papers that are focused on IPD, and we had several variables that we wanted to extract. It's clinical data where we want to see whether patients improve in the 6-minute walking test over time. So we've built a workflow where the agent can take all of those papers, find the right clinical results, extract them as per study extraction, and also using our code execution tool, build pseudo data set of these clinical results and report it. And the whole end-to-end workflow took 15 minutes, and we reached 86.67% accuracy. So maybe now you start seeing what we're trying to do here. If we can combine this deep research with extracting the variables and automatic reporting, we can basically do a meta-analysis, right? So we can extract the right variables from the right papers and automatically summarize them. So this is something we tried recently. For now, we're slightly below 90% accuracy with 1 week of development, so I'm pretty hopeful that it can get even better than that. And there are very many similarities within, how the tools are used within the large language model space. So some of the ideas of what are the differences between Pharos approach and typical ChatGPT approaches. I think especially for organizations, what's important is that if you have proprietary data, we can include it in our knowledge base, and we can dedicate a whole server for only your data, so we can keep it secure. We can also use local models. So if an organization is scared of data leakage, maybe there is something very sensitive there, we're free to use local models, and we're really building around science. So we already vectorized all the publications in cancer research until 1950s, and everything else 5 years backwards. And we're building with biology-specific tools. So this is something that OpenAI maybe wouldn't really think about or wouldn't really find value in. But if a researcher has a tool that they really need to use, we could include it in the platform and let agent use it. And yeah, this is just some of the ideas of what is a wrapper, right? It's a really common term in large language model space. Everything we're building these days is called a wrapper. But there's a lot of development that has to be built around the models in order to make them work. And one example of what people these days don't consider a wrapper, but it's a very common tool, is, for example, Netflix. You know, Netflix is completely built on top of Amazon servers. So we could consider Netflix as being a wrapper for Amazon. And slow wrap up. So we have, we're still early stage, but we have great collaborators. We've completed pilot project at one of the BIA companies, Dandelion, deployed for all employees at Circle. And we have strong academic partners, including one letter of intent from Boston Children's Hospital. And how do we differentiate from different tools? As I mentioned at the beginning, majority of tools within science are very manual, while we want to go as autonomous as possible. Of course, these days the models and technology is not there yet, so I would consider it semi-autonomous with human intervention. But that's where we position ourselves. And we have a great team, so I started the company with Aurel. We both have more than 5 years of experience, me more on the industrial side in biotech companies, while Aurel through his academic career and his postdoc at Danish Cancer Institute. And also we have great developers from top research institutes like Karolinska and University of Cambridge. And we're supported by experts from business and research from top universities like MIT and Harvard. And just to give you an idea, right? So for now, we have a data analytics platform. We're making data analysis faster. We can search through millions of publications that would not be accessible to human in his lifetime. But the idea is greater. So we can see this progression of technology from Garry Kasparov being beat in chess to AlphaFold. And the same progression can be seen within the automation space. So there's much more automation within the laboratories that's happening. There are several cloud labs already. So we hope that with this increase in automation, both on the software side and the hardware side, we can combine both those tools. And the model will not only be able to come up with ideas, but also validation strategies that can be then translated to scripts for, let's say, liquid handlers in the lab to really close the loop of science and deliver life-saving drugs on demand. All right, so thank you for your attention. If you would like to try the platform, then feel free to use the QR code. And we're running a small pilot with a limited number of researchers. And we would be really happy to get your feedback. And if you would like to work with us, then we also look for business and software developers. So yeah, also feel free to scan the QR code. It will get you to our website, to the contact form. But also feel free to approach me afterwards and we can chat about it. So thank you. That's all from me.
Excellent talk. Thank you so much. Let's see if it sparks some questions in the room.
Thank you for a really great talk. That was really interesting. So I was a bit unsure. You gave this example where you said you had 86% accuracy.
Yeah.
Did you give it 5 papers as input? So,
yeah, because, yeah, it's a trial we're doing with one of the partners, but I thought I will give you a kind of glimpse to it. So we knew that those papers will contain the information we want. But the idea is that maybe with combination of the deep researcher, we could find the right papers, right? So you need to optimize a couple of steps there. Yeah. There is a step of finding the right papers, extracting the right variables, and then writing the correct code to analyze them. So this 86 is compared to the ground truth that we manually extracted from the papers. So it's extraction accuracy.
Okay. Yeah, because I was just, so it sounds like you're not really there yet, but what would happen if you were giving it like the full Google Scholar?
Yeah, we will see. Maybe that's going to be next talk.
Yeah. Cool. Thank you.
Yeah.
Yes, we'll definitely invite you back to give a talk when that happens. Any other questions in the room? Yeah.
We have some online. Now, I'm not so much within it, so I try to read out the question as good as I can. So it's from Nikolai Lulasen, and He's asking, in your experience, how hard is it to get LLMs to parse tables and figures out of the box? Do you use any special tracks for OCR on tables, figures?
Yeah, so it's a good question. Basically, with those PDFs, we extracted text data from the PDFs, so there was no OCR necessary. We tried with OpenAI vision model to extract also visually, and it sucked. Yes, that's as much as I can tell now. I think there is a value in this extraction, OCR extraction from the paper documents, but because still many companies use paper. But we didn't try it, so I can't tell you much. But it's pretty good to extract text from text. Text from image, we didn't have good experience.
Thank you.
So how do you see a tool like Site.ai? I didn't see that in your landscape, but they also extract information from scientific literature.
How are they called? Could you repeat?
SCITE.ai.
Yeah, I don't know those guys.
Okay.
But I assume I can make an educated guess that they focus really on extraction, and for us it's building agents. So we want agents to use tools, and extraction could be one of the tools that they will have in their repertoire, right?
Do we have more questions? We do. Did you have one?
Yeah, quick question. So how does your system decide on which hypotheses should be tested? Because a data set could have many different potential hypotheses proposed to be testable by a data set. So
yeah, so that depends on how deep down you want to be. So if you have a really specific question you want to ask, then we can provide an agent with your specific question, and then it will run once and it will try to answer it. But if it's a complex task, it will try many questions until it reaches the right solution. But we don't have prioritization of hypotheses yet. So it's one of the ideas and one of the problems that many researchers told us about. With hypothesis is one, right? But it could be also with you would like to maybe prioritize protein mutants, right? You have a list of maybe many proteins that you optimize, and which one should we pursue next, right? So we haven't looked into it that much yet. For now, the agent gets kind of the free realm. So if we leave it for a number of iterations, it will come up with questions, and by the end summarize them and provide you an answer. But whether it prioritizes the right hypothesis, we still don't know. It's something to look through. It's a good idea.
Yeah, really good question.
Yeah.
Hello, so Daniel from Novo Nordisk, and I sit in drug development also. So very interesting speech. So thanks for that. Yeah. So one thing is around the 10 years. I think we can debate whether it still takes 10 years, but that's one thing. So again, perhaps a little bit on your commercial model. So you primarily look at the research part here and getting the molecules out there. And just a consideration around what's your commercial space, because I think also to the gentleman over there mentioning the saturation in the market. There are many people looking into exactly the space you're in. So just what's your considerations? What's your commercial model to the companies? Are you looking for mid-size or big pharma? And your also, perhaps, I know this is getting a lot of questions, but the last part is around the authorities, because one of the big things in the 10 years or 6 or whatever we are looking at is that when you then talk to the authorities and start to talk about AI, you get into some kind of a squeeze there. So some considerations there would also be nice to have. And sorry for the many questions.
No, very good. I think it's fair to say that we're still figuring out the best sales channel. The whole idea with Pharos started from a very industrial problem. It was not so research-oriented. So the problem was really at the beginning that there is so many databases within big companies. At Novo, you probably faced it pretty clearly. And the Pharos was initially built to build you connectors to all your systems and then ask questions across all of them, right? So that's like a very industrial challenge to solve. But at the end, we want researchers to use it, right? So if we connect the systems, who will at the end analyze this data? Researchers. So that's why we also want to make sure that the analysis part is valuable to the hands-on scientists. So from the perspective, let's say, of a project manager or R&D manager, he can see, all of the questions that researcher asks, all of the analysis and the reports that are fully reproducible. But if the scientists are not satisfied with the answer, then what's the point, right? So it's kind of combining both. The management has the overview of what's happening within the specific team, and the researchers have faster and better analysis.
Thank you very much for a very interesting talk and presentation. My question is still on the quality of the input that you are getting and taking into consideration the meta-analysis as an example. How do you, can you give a little bit more detail in terms of how do you intelligently search? For example, there is a difference between an open label study and a randomized clinical trial. And then how do you assess the methodology was done right, the sample size, and so forth?
So we don't evaluate whether the study was right itself, right? It all depends on the questions. So like the way we did it was that we left LLM with 4 text blocks extracted from those papers, right? And then we provided with a schema of what variables do we want to extract. So you could imagine like one variable was, for example, mean at baseline for a control, right? And you could imagine a schema as a table where there's one variable, and then you have to, like, as a scientist, you would explain what exactly do you mean by that, right? And then the LLM will try to interpret what you mean, so the context, and will look at the paper and extract the value the model thinks is the best, right? But so basically, it's hard to tell whether the model thinks about it correctly. The only way to tell is by comparing it to the ground truth. So that's what we did, right? But it's really based on how well you define the problem as a scientist, right? So for example, we had this discussion with one of our partners about the dropouts in the studies. So we started with, let's say, 10 patients, and then we were left with 8 until the end or something. So how do we calculate? Do we include all 10, even though the measurements are missing or not, right? So this is something a human would have to be really explicit about to extract the right value. Otherwise, the model will select, either of them. Either it will assume we take 10, or it will assume we take 8. So there is still this human and AI interaction where you have to be really precise. The more precise you are, the better variables you get at the end.
Thank you.
And also, just to say, like we just finished this, so it's a week of development. So it's gonna, we will get more data, and then most likely it will be also clearer how to tune those models to get better answers, right?
So we have time for another question. If somebody has a burning question here, yeah.
Hi, thank you. I actually have a startup question. First is, how do you plan to monetize this product? And second is that you mentioned that you got 3 letters of intents. And could you maybe talk a little bit about how you approach and how did you end up with letter of intent?
Yeah, we started as a spin-out from another company, and we already were kind of close to the BII community. So that's where 2 out of 3 letters came from. And then remaining one was through my co-founder, who was a little bit better connected with the Boston's Children's Hospital. And that's how we could present the solution and get the letters of intent. So connections, personal connections, right? That's where we started. Yeah.
How about the monetization model?
Yeah, so we did kind of a short sum up on how big is the opportunity there. And I estimated that there's around 4.5 million researchers out there. And when you look at the solutions that scientists use in many of the big pharma, not Novo yet, I think, a lot of them uses BenchLink. And BenchLink, at least when I was buying it for the company I was working with before, costed $500 per user per month. So that's how we imagine the pricing for the solution for the enterprise partners, right? And that leaves you with a $27 billion market in the B2B SaaS space.
Yeah, thank you.
That's all we're going to have time for this time around. Let's give Bogomil a big hand of applause.
Thank you.
And we'll meet again. Put their QR codes for their LinkedIns up here if you want to follow up with any of the speakers. But I also have a code. I'll just leave it for a sec. We also have a code for the next Applied AI Lunch Talk, which is already happening on April 10th. We're going to be talking about what good AI can do in the world. So it's a little bit of a different focus, but still connected. So I hope to see many of you back here, either physically at Skylab or online. And that's all we have for you for today. Feel free to stick around out here outside of the auditorium, talk to the speakers, talk to each other, or join my colleague Hele, who's going to be doing an open house of Skylab, so you can get a tour and an introduction. Thank you.